Hello, my name is Joshua Gavi. I welcome you to Digestive System. What is digestion? Digestion is the breaking down of large organic molecules obtained from the diet. Examples: the carbohydrates, the fats, the proteins, into smaller ones like glucose, fatty acids, glycerol, and amino acids. How different are intracellular and extracellular digestion? What is the evolutionary advantage of extracellular digestion? Intracellular digestion is that which breaking down of macromolecule takes place within the cell. Extracellular digestion is that in which the macromolecules are broken down in place outside the cell. In extracellular space, in the surrounding of the lumen of the digestive tube. The advent of extracellular digestion is evolution allowed organisms to benefit from greater varieties of food. The breaking down of large molecules into smaller ones outside the cell permitted the use of other foods than those that, due to the size of their molecules, could not be interiorized by diffusion, phagocytosis, and pinocytosis. How is extracellular digestion related to cellular and tissue specialization? A variety of specialized cells and tissue appeared within the extracellular digestion to provide enzyme and special structures for the breaking down of the dietary macromolecules. This phenomenon allowed other cells to be liberated for other tasks and differentiation while benefiting from the nutrients distributed through the circulation. What is the difference between complete digestive system and incomplete digestive system? How are these types of digestive tubes associated and known to extracellular digestion? Animals with incomplete digestive system are those in which the digestive tube has only one opening, like the Cnidaria and Platyhelminths. Animals with a complete digestive system are those in which the digestive tube has two openings. We have the mouth and the anus. All other animal phyla, with the exception of Polyphyla, that do not have any digestive tubes. In animals with incomplete digestive tubes, digestion is mixed. It begins in the extracellular space and finish in the intracellular space. In animals with complete digestive system, extracellular digestion within the digestive tube predominates. What are some evolutionary advantages of animals with complete digestive tube? The complete digestive tube allows animals to continuously feed themselves without waiting for residues to be eliminated before beginning the digestion of new foods. In this way, the absorption of a large amount of nutrient is possible, and therefore bigger and more complex species can develop. Digestive tubes with two openings also makes digestion more efficient, since they provide different sites with different physical and chemical conditions, that is the mouth, the stomach, the bowels, for the action of different complementary digestive enzymatic systems. What is mechanical digestion? In mollusks, arthropods, earthworms, birds, and vertebrates, in general, which organ respectively participate in this type of digestion? Mechanical digestion is the fragmentation of food aided by specialized physical structures, such as the teeth, previously to the extracellular digestion. Mechanical fragmentation of food helps the digestive enzymatic reaction because it provides a larger total area for the contact between the enzyme and their substrate. In some mollusks, the mechanical fragmentation is done by the radula, a teeth-like structure. Some arthropods, like lobsters and dragonflies, have mouth parts that make mechanical digestion of food. In the earthworms and birds, the mechanical digestion is made by internal muscular organs. The mandibulate vertebrates, there are mandibles and chewing muscles to triturate food previously to the chemical digestion. Concerning extracellular digestion, what is meant by chemical digestion? Chemical digestion is a series of enzymatic reaction to break macromolecules into smaller ones. Which type of chemical reaction is the breaking of macromolecule into smaller ones that occurs in digestion? What are the enzymes that participate in this process called? The reactions of the extracellular digestion are hydrolysis reactions. That is to say, breaking of molecule with the help of water. The enzymes that participate in the digestion are hydrolytic enzymes. Which organs of the body are part of human digestive system? The digestive system, also known as Systema digestorium or gastrointestinal system, is composed of the digestive tube organs plus the digestive adrenal glands. Digestive tube is composed of the mouth, pharynx, esophagus, stomach, small intestine, where we have the duodenum, the jejunum, and ileum, and then the large intestine, where we have the cecum, colon, rectum, and the anus. What are peristaltic movements? What is their role in human digestion? Peristaltic is the process of synchronized contraction of muscular wall of the digestive tube. Peristaltic movements may occur from the esophagus until and including the bowels. The peristaltic movements are involuntary. They have the function of moving and mixing food along the digestive tube. Peristaltic movement deficiency, for example, in the case of injuries of the innervation of the muscular wall of the digestive tube, causes Chagas disease, can lead to interruption of the food traffic inside the bowel and to severe clinical consequences like megacolon, abnormal enlargement of the colon, and megaesophagus, enlargement of the esophagus. From the lumen to the external surface, what are the tissues that form the digestive tube? From the internal surface to the external surface, the digestive tube wall is made up of mucosa, that is epithelial tissue, responsible for the intestinal absorption. Submucosa, this is the connective tissue beneath the mucosa membrane and where blood and lymphatic vessels and neurofibers are located. Muscle layers, those are smooth muscle tissue, two layers, one interior circular and the other exterior longitudinal structure responsible for the peristaltic movement. Serous membrane associated with the epithelial and connective tissue forming the external surface of the organ. In the bowels, the serous membrane prolongs to form mesentery. Serous that encloses blood vessels and supports the bowel within the abdominal cavity. What is the location of the salivary gland in the human? There are 6 major salivary glands and they are located in each parotid gland. 2 beneath the mandibles, submandibular, and 2 in the base of the tongue, sublingual. More than 700 other minor salivary glands exist dispersed on the top lip mucosa, gingiva, palate, and the pharynx. What is the approximate pH of the salivary secretion? Is it an acidic or basic fluid? What is the main function of saliva? The salivary pH is approximately 6.8, and this thus slightly acidic pH. Saliva lubricates the food bolus and initiates enzymatic extracellular digestion of food. It works as a buffer for the mouth pH. It has an important role of having IgA1 antibodies, also present in tears, colostrum, mother's milk, and in the mucosa of the intestine and airways, that protects the organism against pathogens. What is the salivary digestive enzyme? Which type of food does it digest, and into which smaller molecules does it transform the food? Salivary hydrolase is also known as salivary amylase or ptyalin. Ptyalin digests carbohydrate, breaking down starch and glycogen, glucose polymers into maltose, a glucose disaccharide, and dextrin. Why doesn't the food enter the trachea instead of going to the esophagus? When food is swallowed, the swallow reflex is activated and the larynx elevates and closes to avoid the portion of food bolus from entering the trachea, causing aspiration of strange material to the bronchi. Is the esophagus a muscular organ? Why, even in the patient lying totally flat on the hospital bed, can swallowed food reach the stomach? The esophagus is predominantly a muscular organ, so the assertion is correct. The esophagus is a muscular tube formed of its superior third with striated muscle tissue, its middle third mixed muscle tissue, striated and smooth, and its lower third smooth muscle tissue. The peristalsis of the esophagus provides the movement of the food towards the stomach even without gravitation help. What is the route of the ingested food from swallowing into the duodenum? Until reaching the duodenum, the food enters the mouth, passes the pharynx, goes down the esophagus, and passes the stomach. What is the value that separates the stomach and the esophagus called? What is its function? The valve that separates the stomach from the esophagus is the cardia. Its function is to prevent acid gastric content from entering back into the esophagus. Insufficiency of this valve causes gastroesophageal reflux, a disease in which complain by patients of bloating or heartburn, retrosternal burning. What is the valve that separates the duodenum from the stomach wall, and what is its function? The valve that separates the stomach from the duodenum is pylorus. Its function is keeping the food bolus within the gastric cavity for enough time to allow gastric digestion to take place. It also has a function of preventing intestinal content from going back into the stomach. What is the pH inside the stomach? Why is there need to keep that pH level? How is it maintained? Which are the cells that produce that pH? The normal pH of the gastric juice is around 2, so it is an acidic pH. It is necessary for the gastric pH to be kept acidic for the activation of pepsinogen, a pre-enzyme secreted by gastric Schwann cells, into pepsin, digestive enzyme that acts only under low pH. This pH level is attained by secretion of hydrochloric acid by parietal cells. Besides being fundamental for the activation of the main gastric digestive enzyme, how does HCl also directly participate in digestion? With its corrosive effect, HCl helps to rupture the adhesion between the food particles, facilitating the digestive process. How is the gastric mucosa protected from the acidic pH of the stomach? The gastric epithelium is mucus secretory, that is, it produces mucus. The mucus covers the stomach wall, preventing corrosion by the gastric juice. What is the digestive enzyme that acts within the stomach? Which type of food does it digest? What are the cells that produce that enzyme? The digestive enzyme that acts in the stomach is pepsin. Pepsin has the function of breaking down proteins into smaller peptides. The gastric cells that produce pepsinogen, the enzymatic precursor of pepsin, are the Schwann cells. What name does the food bolus that passes from the stomach to the duodenum get? The partially digested and semi-fluid bolus that leaves the stomach and enters the duodenum is called chyme. What are the three parts of the small intestine? The small intestine is divided into three portions: the duodenum, jejunum, and ileum. By generally dividing food into carbohydrates, fats, and proteins, and considering digestive process until pylorus, that is exit of the stomach, which of these mentioned types of food have already undergone chemical digestion? Until the exit of the stomach, carbohydrates in the mouth and proteins in the stomach have already undergone chemical breaking down by digestive enzymes. Carbohydrates have suffered the action of salivary amylase, ptyalin. Proteins have suffered the action of enzyme pepsin, the gastric juice. Fats, until reaching the duodenum, do not undergo chemical digestion. What is the substance produced in the liver that acts on small intestine during digestion? How does that substance act in the digestive process? Bile, an emulsifier liquid, is made in the liver, later stored in the gallbladder, and released in the duodenum. Bile is composed of bile salts, cholesterol, and bile pigments. Bile salts are detergents, amphiphilic molecules, that is, molecule with the polar water-soluble portion and non-polar fat-soluble portions. This feature allows bile salt to enclose fats inside the water-soluble micelles, and the process is called emulsification, for them to be in contact with intestinal lipase, enzymes that breaks fats into simpler fatty acids and glycerols. What is adrenal organ of the digestive system in which bile is stored? How does the organ react to the ingestion of fat-rich food? Bile is concentrated and stored in the gallbladder. When fat-rich food is ingested, the gallbladder contracts and releases bile inside the duodenum. This is the reason why patients with gallstones must not ingest fatty food. The reactive contraction of the gallbladder may move some of the stones to the point of blocking the duct, and it drains bile into the duodenum, causing pain and possible severe complications. What is the digestive function of the liver? Besides making bile and releasing it into the duodenum, the liver has other digestive functions. The venous network that absorbs nutrients from the guts, called mesenteric circulation, drains its blood content almost entirely to the hepatic portal vein. This vein irrigates the liver with absorbed material from digestion. So the liver has the functions of storing, processing, and inactivating nutrients. Glucose is polymerized into glycogen in the liver. This organ also stores many vitamins and iron absorbed in the intestines. Some important metabolic molecules like albumin, clotting factors are made in the liver from amino acids of the diet. The liver ingested toxic substances like alcohol and drugs are inactivated too. Beside the liver, which is the other adnexal gland of the digestive system that releases substances in the duodenum participating in extracellular digestion. The other adnexal gland in the digestive system is the pancreas. This organ makes digestive enzymes that digest protein, protease, lipids, lipase, carbohydrates, pancreatic amylase. Other digestive enzymes are gelatinase, elastase, carboxypeptidase, ribonuclease, deoxyribonuclease are also secreted by the pancreas. How does the pancreatic juice participate in the digestion of proteins? What are the involved enzymes? The pancreas secretes trypsinogen that, undergoing the action of enzyme enterokinase secreted by the duodenum, is transformed into trypsin. Trypsin, in its turn, catalyzes the activation of the pancreatic chymotrypsin into chymotrypsin. Trypsin and chymotrypsin are proteases that break down proteins into smaller peptides. The smaller peptides are then now broken down into amino acid by the enzyme carboxypeptidase, also secreted by the pancreas in a zymogen form and activated by trypsin, helped by the enzyme aminopeptidase made in the intestinal mucosa membrane. How does the pancreatic juice resume the digestion of carbohydrates? What is the involved enzyme? Carbohydrate digestion begins with the action of salivary amylase or ptyalin in the mouth, and it continues in the duodenum by the action of pancreatic juice. This juice contains enzyme pancreatic amylase and amylopsin that breaks down starch, amiram, into maltose, a disaccharide made up of two glucose molecules. How does the pancreatic juice help the digestion of lipids? What is involved enzyme? The enzyme pancreatic lipase present in pancreatic juice. This enzyme breaks triglycerol, triglyceride into fatty acids and glycerol. Beside the pancreatic juice, in the intestine, there is a releasing of enteric juice that contain digestive enzymes too. What are these enzymes, and which type of molecules do these enzymes break? Enteric juice is secreted by the small intestine mucosa. The enzyme in the enteric juice and their respective function are described below. Enterokinase, enzyme that activates trypsinogen to trypsin. Saccharase, enzymes that breaks down sucrose into glucose and fructose. Maltase, enzymes that break down maltose into 2-glucose molecules. Lactase, enzymes that breaks down lactose into glucose and galactose. Peptidase, enzymes that breaks down oligopeptides into amino acids. Nucleotidase, enzyme that break down nucleotides into its component nitrogen-containing bases, phosphates, and pentase. Coming from the acid pH of the stomach, which pH level does the chyme find when it enters in the duodenum? Why is it necessary to maintain that pH level in the small intestine? What are the organs responsible for that pH level, and how is it kept? Entering the duodenum, the chyme meets the pancreatic juice under a pH of approximately 8.5. The neutralization of the chyme acidity is necessary to keep adequate pH level for the functioning of digestive enzyme that acts in the duodenum. Without the neutralization of the chyme acidity, the mucous membrane in the intestine would be injured. With the stimulated by chyme acidity, duodenum makes the hormone called secretin. Secretin stimulates the pancreas to release the pancreatic juice, and also the gallbladder to expel bile in the duodenum. The pancreatic secretion, rich in bicarbonate ions, is released in the duodenum and neutralizes the chyme acidity. This acidity is neutralized by the secretion of bile in the duodenum lumen. What are the 5 human digestive secretions? Which of them is the only one that does not contain digestive enzymes? Human digestive secretion are saliva, gastric juice, bile, pancreatic juice, and enteric juice. Among these secretions, only the bile does not contain digestive enzymes. Why do proteases supplying cells of the stomach and the pancreas make only precursors of the active protein enzymes. The stomach and the pancreas make zymogens of the protease, pepsin, chymotrypsin, and trypsin. And these zymogens are released into the gastric or duodenum lumen for activation. This happens to prevent the digestion of these organs, the stomach and the pancreas, own cells and tissues, by the active form of the enzymes. So, the production of zymogens is a protective strategy against the natural effect of the proteolytic enzymes. After digestion, the next step is absorption done by the cells of the mucous membrane of the intestine. For this task, large absorption surface is an advantage. How is it possible in the small intestine space of the body of the pluricellular organism to present a large intestinal surface? Evolution tried to solve this problem in two ways. The simplest is the long tubular shape of the bowel, approximately 8 meters in extension, making possible the numerous small intestine loop folded closely. More efficiently, solution are the intestinal villi and the microvilli of the mucosal membrane cells. The intestinal wall is not smooth. Mucosal membrane, together with its submucosa, projects inside the gut lumen like glove fingers, forming invagination and villi that multiply the available surface for absorption. In addition, the epithelial cells that cover these villi have themselves numericella-like projection called microvilli on the external surface and their plasma membrane. The absorption area of the intestine of these increase the hundreds of times of these solutions. In the jejunum and the ileum, there are folds that have the function of increasing absorption surface too. In which part of the digestive tube is water chiefly absorbed? What about amino ions and vitamins? Most part of water, vitamins, and amino ions are absorbed by the small intestine. Large intestine, however, is responsible for reabsorption of nearly 10% of ingested water, an important amount that gives consistency to feces. That is, the colon disease can cause diarrhea. From the intestinal lumen through the tissue, what is the route of nutrients after digestion? Monosaccharides, amino acids, mineral salts, and water are absorbed by the intestinal epithelium and collected by capillary vessels of the intestinal villi. From the capillaries, nutrients go to the mesenteric circulation, a system of vessels that drains the intestinal loops. The blood of the mesenteric circulation is drained in the portal hepatic vein, and some nutrients are processed by the liver. From the liver, nutrients are gathered in the hepatic veins that discharges its blood content into the inferior vena cava. Blood from the inferior vena cava then gains the right chamber of the heart and is pumped to the lungs for oxygenation. From the lungs, the blood is returned to the heart where it is pumped to the tissue, distributing nutrients and oxygen. What is the special route that lipids follow during digestion? What are chylomicrons? Triglyceride emulsified by bile within the micelles suffer the action of lipase that breaks them into fatty acids and glycerol. The fatty acids and glycerol and cholesterol are absorbed by the intestinal mucosa. In the interior of the mucosa cell, fatty acids and glycerol form again the triglyceride that together with the cholesterol and phospholipids are packed in small vessels covered by proteins and called chylomicrons. The chylomicrons are released in the minuscule lymphatic vessels, not in the blood vessels, and they gain the lymphatic circulation. So the lymphatic system plays an important role in the absorption of the lipids. The lymphatic circulation drains the content in the venous blood circulation. In that matter, cyclomicrons reach the liver, where their lipid content is processed and released in the blood under the form of protein-containing complexes called lipoproteins, like HDL, VDL, LDL. What are the so-called good and bad cholesterol? Lipoproteins are complexes made of lipids, triglycerides, and cholesterol, and proteins. Lipoproteins present different densities according to the relationship between their protein and lipid quantities. Since lipids are less dense than protein, low-density lipoproteins, LDL, are those with low protein-lipid relationship. High-density lipoprotein have high protein/lipo relationship. Another group is very low-density lipoproteins with very low proteins/lipid relationship. LDL is also known as bad cholesterol because it transports cholesterol from the liver to the tissues and so induces the formation of atheroma plaque inside the blood vessels, a condition called atherosclerosis that can lead to severe circulatory obstruction like acute myocardial infarction, cerebrovascular accidents, and thrombosis. HDL is also known as good cholesterol, since it transports cholesterol from the tissue to the liver to be eliminated with bile. Elevation of HDL blood level reduces the risk of atherosclerosis. VLDL transformed into LDL after losing triglycerides in the blood. Why does the ingestion of vegetable fibers improve the bowel habits in people that suffer from hard stool? Some types of plant fibers are not absorbed by the intestine, but play an important role in functioning of the organs. They retain water inside the bowels and thus contribute to the softening of the fecal bolus. The soft fecal bolus is easier to be eliminated during defecation. People that eat less dietary fiber may suffer from hard stool and constipation. What are the main functions of the bacterial flora within the human gut? Bacteria that live in the gut have a great importance in digestion. Some polysaccharides like cellulose, hemicellulose, and pectin are not digested by the digestive enzymes secreted in the body. Instead, they are broken down by enzymes released by the bacteria of the gastrointestinal tract. The intestinal bacteria flora also makes vital substances for the functioning of the bowel, facilitating or blocking the absorption of nutrients and stimulating or reducing peristalsis. Some gut bacteria are the main source of vitamin K for the body, and they are essential for the blood clotting process. The intestinal flora, they are utile, but also potentially harmful bacteria. It is estimated that more than 100 trillion bacteria live in the human gut. So, some bacteria are useful too because they compete with other species, preventing excessive proliferation of these bacteria. The releasing of digestive secretion is controlled by hormones. What are the hormones that participate in this regulation? The hormones that participate in the regulation of digestion are gastrin, secretin, cholecystokinin, and enterogastrone. How is it produced, and what is the function of gastrin in the digestive process? The presence of food in the stomach stimulates the secretion of gastrin, and in its turn triggers the releasing of the gastric juice. Where is it produced, and what is the function of secretin in the digestive process? Secretin is made in the duodenum. Chyme acidity causes the duodenum to release this hormone, and in its turn stimulates the secretion of the pancreatic juice. How is it produced, and what is the function of cholecystokinin in the digestive process? The fat level of the chyme detected in the duodenum stimulates the secretion of cholecystokinin, CCK. CCK acts by stimulating the secretion of the pancreatic juice also and the releasing of bile by the gallbladder. Where is it produced? What is its function of the entellogastron in the digestive process? When the chyme is too fatty, there is secretion of entellogastron in the duodenum. The hormone reduces peristalsis of the stomach and also the entrance of food into the duodenum, as the digestion of fats takes more time. What are the special structures of the avian digestive tube and their respective functions? The digestive tube of bird has special structures. In this sequential order: the crop, proventricus, and the gizzard. The crop has a function of temporary storage of ingested food, and it is more dilated area of the avian esophagus. The proventriculus is the chemical stomach of the bird, where food is mixed with digestive enzymes. The gizzard is the masticular pouch that serves as the mechanical stomach, where food is ground to increase the exposure area of the food particles for the digestive enzymes to act. Compared to mammals, do birds absorb more or less water in their digestive system? Why is this phenomenon an adaptation to flight? Bird feces are more liquid than mammal feces. That is, less water is absorbed in the avian digestive system. The more frequent elimination of feces in bird is due to their less solid feces, which is an adaptation to flight since their body weight is kept lower. What is meant by mutualistic exploration of cellulose digestion? A phenomenon that occurs in some mammals and insects. Herbivorous animals eat great amount of cellulose, a substance not digested by the digestive enzyme. In these animal, regions of the digestive tube are colonized by microorganisms that digest cellulose. The mutualistic ecological interaction between animals and microorganisms occurs, for example, in horses, cows, rabbits, and some insects such as the termites. Cows swallow the food once, and then the food goes back to the mouth to be chewed again. How can this phenomenon be explained? The food ingested by the cow and other ruminant animal passes first within the two compartments of the digestive tube, called the lumen and the reticulum. Within the food suffers the action of digestive enzyme released by microorganisms that live in a mutualistic ecological interaction. The reticulum of the food is divided into some food boluses too. After passing the reticulum, the food card is regurgitated to the mouth to be again chewed and swallowed in the process called lumination. The digestive food that enters the omasum, where it is mechanically mixed after the food goes to the abomasum, the organ where chemical digestion takes place. After leaving the abomasum, the true stomach, the food bolus gains the intestine. Thank you very much for participating in Digestive System. Please do like, share, and subscribe.